I come back into focus here.
Oh, okay.
Hello.
Hello. Hello, Morgan.
Good morning.
Good morning. So, how's everyone today? How is Susan we were talking about your project on for the last several weeks. So
Well, I'm still working on it, but I found tiling that would go in a square box for the sides of both the triangle prism and my hexagon configuration, and it goes straight down and up instead of twisting all over the place. So I've got something, and maybe I can say, well, we can try all these other configurations too. So I filled out that section. I just need to do the hexagon model.
yeah.
Maybe in a couple of days I'm going to write up the triangle and some of the other things and make sure my essay's together, paper, whatever.
Yeah.
Yeah. Yeah, it's going to be a 10,000 word paper.
Oh, okay. Yeah, that's right. Right about like a very nice long publication.
Yeah. This is what cells do. So this is what I've done to try to model them. Yeah.
Yeah. Well, that's great. Good luck with that. Yeah.
Yeah. Well, at least it goes straight up and down. It runs into itself, but I knew that would happen. So.
Yeah. Great. Morgan, how are you?
I am just getting things ready here for the school run. Yeah, just well, I'll be listening in and happy to hear what you guys are talking about.
Yeah, great. Hi, Jesse. How are you? Good. Yeah. Hi. So yeah, I guess I don't know if Dick will join us today. He asked about like our progress on the paper that we're working on for the sort of the molecular biology of differentiation, and we're still working on revising that. It'll take a while, but I think we'll get it out hopefully by the end of the year. But it's actively being worked on. So just have to kind of incorporate some comments and rethink some things, and so that's good. Yeah. So we've finished up with our GSOC stuff, and we have Sushmanth. He's busy right now. He'll be rejoining our group soon, next couple weeks. And then I know that Himanshu had wanted to continue on. I don't know. He's been having his own issues over the summer, so we hopefully if he wants to join in, he's welcome to. Otherwise, I'm not going to really go over any of the repositories today, but we have to kind of keep on the treadmill with this because the new GSOC projects are, they want to see proposals at the end of January. So,
oh wow,
kind of comes sooner than you'd like. So, I'm thinking about continuing some of the projects that we were working on this summer, or maybe some of the projects we were working on last year.
Yeah, and I'm sorry I haven't got you those images of the eggs yet. They're more fuzzy than flipping microscope eggs.
Yeah.
Images, because the cameras aren't as good. And I took a whole bunch of the flipping microscope images And so I was able to choose ones that were in focus. So, yeah.
Yeah.
Anyway, just haven't got there. I need to finish this project.
Oh yeah, definitely.
My paper. They want me to present this early, either late December or mid-December or early January. So, whichever.
Yeah.
Anyways, yeah.
Well, that's good.
Anyway, I don't know.
Yeah. I mean, I've talked to Hari Krishna recently about some of the things he's been wanting to build, like a NERF model, which is a neural radiance field model. And I guess he needs, more data ideally, but I don't know what his progress is on that. So we'll see. I mean, I haven't heard from him in a while on it. So
if someone, I've asked 2 people for eggs. I had a death of one. I had Calgary died. Poor Calgary died. My salamanders. The water in Morden is awful.
Oh yeah.
And where I get the water from, the reverse osmosis water, is under a boiled water advisory at the moment. So it's like, yeah, well, I had a salamander die because of your water. Just like, anyways, I guess I need to search Kijiji for more salamanders.
Yeah.
Anyway, I have a new one. I'm calling her Lucy. So.
Oh yeah.
Because they leucistic Lucy.
Oh, okay. It's always, it's always a thing, like with biology experiments and biology setups, that, if your water is of different quality, it can have an effect on the experiments. So, like, sometimes if you just like have like a really bad part of the year in terms of pollen or something, you can have like a lot of. I remember when we used to work in the lab in cell culture, every year in the fall and spring, we'd have problems with like residue, or like getting getting our cultures infected because there was all sorts of stuff floating around the air and just happened to make it into the culture. And so your odds of your experiment not working at that time were much greater. And like water, if you don't sometimes the source water just doesn't agree, even if you filter it. It's just still.
Yeah, well, it's reverse osmosis water, but I swear they hadn't fixed their filters recently or something.
Yeah.
I get the reverse osmosis for my house, and they tend to just drink the stuff that I've boiled.
Yeah.
This is ridiculous. I just don't trust it because my salamanders died. Yeah, I lost Calgary. Like, she was sick.
Oh.
Anyway, like, she wasn't that very well.
Yeah.
To begin with, and yeah. Anyways, yeah.
Yeah. Hello, Dick. How are you?
Hello. Okay.
How are things going with you?
I once tried sterilizing water with a UV sterilizer, and I was raising zebrafish, and they wouldn't breed. Turned out the UV sterilizer heated a copper pipe, driving copper into the water, which made them sterile.
Oh no.
Yeah. So too much of a good thing.
Yeah.
Okay.
Yeah.
Anyway, it's hard to win on these problems with water quality.
Yeah. Yeah.
Well, Morden water tastes worse than Winnipeg water. If you know what I mean.
Yeah.
So, anyways,
no iron deficiency if you drink Alonzo water.
Oh, okay. Yeah. Yeah, well, you'll get a good boost to your immune system if you drink our well water.
Well,
You get
that's what
sick for a few days, and then your immune system picks up. Yeah. Oh well. I'm yeah.
Yeah.
Don't know. At any rate, hopefully I'll eventually be allowed to show my advisors what I would like to do for an experiment, which is just to image the salamander eggs with UV, not UV, infrared light, like the optical coherence tomography system.
Right.
Anyway, I've got a whole bunch of slides. I think I showed them to you.
Yeah.
Maybe did I show them to you? Yeah.
Yeah, I think so.
Yeah. So if I finish this 10,000 word essay in good time, I can and present it. Then I can show them this next step, perhaps.
Yeah.
But even if I don't do this experiment, like somebody should.
Yeah.
Yeah.
Yeah, that's great. Yeah, you can see my screen. So actually, yeah, Susan and I have been talking about these. I guess 2 weeks ago we talked about how you have, this apical thing going on with her model. Where it kind of goes up. I don't remember too much about it other than that right now. But basically, this is a paper on optogenetic control of apical constriction induces synthetic morphogenesis in mammalian tissues. So this isn't epithelial cells necessarily. This is actually it is epithelial cells. I'm sorry, and they're using synthetic biology techniques, specifically with respect to optogenetic control, and they're kind of looking at this aspect of the tissue, an epithelial tissue in the kidney. So this tool. I think they're going to talk mainly about this tool OptoSherm3, which is an optogenetics tool. So an optogenetics it works by if you have a light source and you illuminate a cell, you can trigger different genes to be expressed in that cell. So it carries a marker that allows it to be turned on and off by this light pulse. And so there are different types of optogenetic tools that they have out, different types of light source that you can use. So there's this sort of control over like gene expression, and they use this in organoids. They also use this in other tissues. I think they can actually do it on live and behaving animals. So this is not something that this is something that's pretty diverse and can answer a lot of questions potentially. So let's see, we have a comment. Oh yeah, Morgan says there was a great video released by the CC3D dev team. So this is CompuCell 3D, Morgan. Yeah. Yeah, if you could share the link in the chat, we can talk about it after this.
Yeah, I'm just finding the link right now.
Yeah. So yeah, this is so this paper is talks about synthetic biology and how it proposes bottom-up approaches to examine the contribution of each cellular process to complex morphogenesis. So they have this, they want to investigate morphogenesis using optogenetics. It's hard to find tools to manipulate the three-dimensional shapes of mammalian tissues. So, we know that from some of the stuff we've read that, sculpting tissues is possible, but, it's hard to find tools to do this. So what they're going to do is they're going to use an optogenetics tool to sort of shape what they call fast spatiotemporal control of apical constriction. So this is where this apical constriction phenomena and development is going to be under this fast spatiotemporal control, and they're going to look specifically at mammalian epithelia. So this is in epithelial cells. It looks like kidney cells. They're going to have, I guess they're doing this in culture, where they have a sheet of MDCK epithelial cells, and they do these experiments. So they use this tool OptoShroom 3, which is the name of it. I'm not familiar with it before reading this paper, so I don't know too much about it. But this, when they activate this through illumination and epithelial cell sheet, this reduces the apical surface on the stimulated cells and causes displacements in the adjacent regions. Maybe they'll have some pictures later on. We can see light-induced apical constriction provokes the folding of epithelial cell colonies. So this is something that when you get apical constriction, when you can induce it with light, you can fold the cell colony, the cell sheet, and they're working on a soft gel substrate. So they're working on the substrate, they're folding the cells, they're doing this in a controlled way. Its application to marine and human neural organoids. So they're actually interested in organoid models here. Its application to marine and human neural organoids, so mouse, rat, and human, leads to thickening of neuroepithelia, apical lumen reduction in optical vessels, and flattening in neuroectodermal tissues. And so they've been able to do this in organoids where they have these different structures that are forming. They're able to manipulate those structures. These results show that spatiotemporal control of apical constriction can trigger several types of 3D deformation depending on the initial tissue context.
A little bit of confusion here. Did I hear you correctly that the optogenetic apical constriction can lead to flattening of cells?
Why don't we go back? Let's go back to the paper. I'm not sure. Let's see. Well, what they were doing in this paper was that they were using optogenetics. They were on this substrate of some sort of gel, and then they had this. They were able to fold the sheet of cells. So this provokes the folding of cell colonies on soft gels. And I mean, later in the paper, I'll talk a little bit more about it, but I'm not sure if it
did they or did they not say flattening?
Yes, optical vessels flattening in neuroectoderm tissue.
Where is that?
The bottom line, bottom 2, 3 up.
3 up.
That's counter.
Oh yeah, the flattening. Yeah. So they did thickening of neuroepithelial and then flattening in neuroectodermal tissues. So these are in this is in a organoid model. So this is where they have. I'm not sure how they're configured.
It doesn't make sense if you constrict an a cell apically, it should get taller, not flat.
Yeah. Yeah, I don't know why that would be. Huh.
Yeah. Okay. Yeah. Is there an illustration of flattening?
Well, let's see. Yeah, that's what I wanted to look at. So let's see if this is kind of the. Okay, so this shows apical constriction. This is the apical basal. So this is what actually I illustrated for Susan 2 weeks ago, where we had the cell that was hexagonal and it had the apical and the basal part, and then the apical constriction was at the top. It was kind of pulling up like that, and then this is just showing the sort of the validation of the optogenetics.
This is if it goes around a curve, it will do that.
Okay.
Yeah, the whole sheet should curve then.
Yeah.
But, the sheet will curve.
Okay. When Jacobson and I observed apical constriction, the shape was not this conical shape. It was a tall, long cylinder.
I suppose it depends on how the bottom and the top of the cell relate to each other.
We also observed microtubules which went from top to bottom, and they might be involved.
Yes, like in some cells there's microtubules on the sides of cells, according to a couple of things I have.
You can burn the cytoplasm. That's okay.
Anyway, my model actually has the microtubules from.
Yeah, but I don't see how flattening occurs.
How flattening occurs?
Yeah, why? If you constrict the apical end, how on earth do you get flattening?
I think you would get curving.
Yeah, curving is not flattening. I agree.
Yeah.
Yeah, I don't know, unless you've got like a pseudo-stratified epithelium.
Oh, okay, multi-layered. Okay.
Yeah, then
Maybe, but
the cell next to it would be the opposite shape. So then it would be flat.
Yeah, it seems we need clarification of whether they got real flattening or not.
Yeah, well, this depends. Like there are so many variables with this stuff. It's like that's why it took 10,000 words to describe it. It's like, yeah, where do you start and begin with it? Start and end.
Yeah.
I don't know. Yeah.
So this is just, I think, probably just the validation of the technique. So let's see if they have any images of like what they were doing in the. So this is actually a simulated version of this sheet of tissue. So these are the epithelial tissues, and it's a sheet. So this is the sort of the squares that they have here, or that they're just simulating the cells. So they're stimulating at a different time in different time courses. So there's group stimulation, and then they have a 30-minute stimulation, and then they have it where they turn it off and on for like, I think probably about 120 minutes, so about 2 hours. That's 2 hours of stimulation, and then they have this time course to 230 minutes. And they show kind of the effects on the sheet of cells, not just the single cells here, where they have, let's see if I can zoom in a little bit. So this is where they have the sheet of cells, and they're kind of showing the deformation in the sheet here. They have kind of an example here. And then this is the cell where the apical area is here, the basal area is here. So the apical area is smaller than the basal area during this transformation. So as they're stimulating the cells with light, the cells change their shape gradually. And they show this after, so 30 minutes and 150 minutes in green. So the 30 minutes in purple, or whatever that is, it's a different shape. You can see they're overlaying the two images. And so there's a shift in the cell walls in these two images. So you see here where it's kind of like in this direction, up and to the left, and then this 150 minutes it's up and to the right.
Just so long as my dogs don't start barking.
Yeah.
In response.
So yeah, and then so this is what is this image here? This is where we have apical displacements on confluent monolayers. So this is where you have this displacement. They just show the displacement. So it's basically as the shape is changing, the entire sheet is changing its orientation, and things are shifting around. So you have the stimulated area in the middle, you have the adjacent areas outward, and then the outer area beyond that. And so you can see from here that the displacements change as you go out from the stimulated area. And that's all just to show that this technique works. And so and then they show this figure here, which is figure 3. Actually, I think this is part of figure 2. I'm not sure. This is figure 3, but this is not really labeled here. Here it is, figure 4. So there's you can also induce colony folding, and so they talk about flat MDCK layer monolayers in a thick matrigel bed. They have the different images of this colony-induced folding, or this induced colony folding. So you see here you have curvature along the edges. This is the projected area. So you have curvature at the edges of the projected area. So they're projecting light onto the sheet of cells, and the edges kind of curl up off of the matrigel, as you can see here. So they have this glass substrate, they have the matrigel substrate, and then they have the cell sheet that's on top of it, and it's curving up at the edges of the stimulated area. And so you can see that they're showing how it can change its shape. It curls up basically in the apical direction, I guess, after 24 hours. So at 0 hours, it's kind of flat. 24 hours, it's curling up like this. So you see this curvature there. And so I don't know what, if they mean by flattening, they mean that it stays on the matrigel, or if it's curling up versus not curling up, or what they mean by that. It could be an artifact of the way they're doing these experiments. So let's see. So a possible explanation for these differences is the specific tissue geometry and availability of cytoskeletal components in each cell type, as well as the interactions of the extracellular matrix. So optoSHRM3 activation provokes the recruitment of non-muscle myosin 2B to the apical junctions. Although it has been reported that SHRM3 can induce a redistribution of tubulin to cause cell elongation along the apical basal axis, we did not find a change in the distribution of microtubules and NDCK cells or optical vesicle organoids upon optoSHRM3 activation. So so based on their 3D cell segmentation and their model that they're using for modeling some of this, it suggests that optoSHRM3 induced apical constriction could itself be enough to explain cell elongation or cell volume conservation consistent with previous studies. And so, yeah, this is, and then this is the final figure, I think. This shows the mouse optical vesicle organoids. So these are vesicle organoids for the optic cup, I guess. So this is day 0. You have it sits on matrigel. This is day 4. You get this neuroepithelium start to form. It goes off and it starts to thicken and have like this apical and basal aspect to it. And then day 6, it forms this shape here where it forms optical vesicles at the edges of this sphere, and then you get shrinkage within each of these vesicles and an apical and a basal aspect to it as well after day 6. So this is kind of showing this how this is their experiments in the organoids. This is the imaging of the organoids. This is one of the vesicles here. This vesicle shows up. They kind of zoom in on it here. They show the optical stimulation. They show sort of before stimulation, which is in purple. So there's a certain shape before stimulation. Then after stimulation, there's a shift in the shape. So if you see this semicircle right here in the vesicle, the purple at the edge is where it starts. And then after 55 minutes, it kind of contracts down to where this is here. So you see the green semicircle. So there's a shift in the shape over that time. This shows human neuroectodermal organoids. So this is day 0 where you get this, have this human stem cell aggregate or pluripotent cell aggregate. This is day 0. At day 5, you get neural induction. You get the basal and apical aspect. And then day 6 to 7, they say there's flattening. So what they mean by flattening here is where you stimulate the cells in this as neural induction is happening at day 5, you get this flattening of this shape. So you get a flattening in the sheet in this orientation along the edge of this neuroectoderm. So you see the flattening instead of it being this. Yeah, so this shows in a little bit more detail. The surface curvature decreases as the neuroectoderm is stimulated. And then this shows the expected curvature before stimulation and after stimulation. So this is curvature in microns, power of -1. So it's like a fraction of curvature, then orientation, or a fraction of microns, and then orientation in degrees. So this is a very small number here. This is degrees in orientation, so along this curve. And this is, so this is 150 degrees, negative 150 degrees. So you're looking at about 300 degrees of this orientation. And then you have this where there's the stimulation in this blue bar here. And you can see that that's where most of our change is happening in the shape, in the curvature, where the curvature is decreasing. So for example, before stimulation is the blue line, which is pretty much, around this mean curvature, inverse curvature of 0.006. And then you have the red, which is 1 hour of stimulation. So you see after 1 hour of stimulation, the area that's not being stimulated is more or less consistent with what happened, what the shape was before stimulation. There's some shifting of the sheet, here and there, but where you see the treatment, where this stimulation is happening, where the light is being shined in this very small sort of aperture, you get this big difference in 1 hour of stimulation. The curvature decreases, I guess, significantly. They don't really define what that means, but you see this big departure from what was happening before stimulation. So again, you get this flattening that happens as a result of stimulation, this flattening of the epithelial sheet. It's kind of pushing, inward, or it's flattening or something like that. And you see it's a product of the light stimulation and not just happening at random. So that's all they have really, maybe of interest here. They used IPS cell lines for their inputs. So these are, I don't know if that's going to make a difference in terms of, they use the MDCK cell line for a lot of these experiments, but they also use mouse ES cells and human IPS cells. So okay, this is the link to the CC3D stuff. This is the YouTube video, and then this more relates to software engineering of complex computational modeling projects like CompuCell 3D. Yeah, well, Morgan and I have been talking about CompuCell 3D, kind of in some conversations. So yeah, this is something we tried to do a while back, but it's, I don't know, maybe the learning curve is a bit high for some people to learn how to use it. But Morgan, I don't know what your thoughts are on that. I think you need to kind of, if we want to do this, we have to kind of develop a way to, get people ramped up and, maybe try some, because they have a lot of example model systems.
Right. But they've got great worked, embryology examples.
Yeah, they do.
And it's really just getting through. Well, any project with it is very much a software project.
Oh, yeah.
Like a software development project, and maybe a little bit more than just a scripting project. If you see what I mean.
Yes, I know.
But it was. This was what was nice about this video was that it seemed like somebody, took an hour and 45 minutes to go through how to work with the code base and and with GitHub, and sync with their development tree. It was nice.
So is that CompuCell 3D?
Yeah. This is it James Glazer, or leads that at Indiana University, and he does he does obviously a lot of biology, but also network dynamics, and very very multidisciplinary.
I just wondered if it was something I should include in modeling of cells, because I have to write a section, I still haven't, on how to do modeling with tensegrity of cells. And so there's some stuff done in that lab. And I don't know.
I think he's like one level. I mean, it's a lot about the simulations. Just looking across like his student simulations, they're usually not so much, you know, of the finite element sort. Although they talk sometimes about integrating that, but very much like what are simplifying assumptions we can make to deal with either multicellularity and or, you know, they do pandemic modeling, you know, they do you know, very generic modeling as well, right? So you know, infection and things like that, where things are transmitted and passed, you know, passing through barriers. And I'm being really generic because that's the kind of stuff, you know, whether it's like tissue modeling as well as, you know, population modeling. Does that make sense?
Okay.
But he does do. I mean, he definitely does do. Yeah. So it's just if I was modeling a cell, again, like yeah, I just need to know what the problem was. But you know, like the 3D seems to be in its name, or the kind of, you know, spatial aspects of multicellularity, like seem to have been an early development goal.
Okay, I need to look at this then.
Oh, okay. Yeah, I mean, it's like, if it's embryology, like, he's definitely got some great worked examples. I'll find some things I've been looking at. Yeah.
Well, that's the biology that I know. Embryology. Thank you, Dick. I know normal physiology as well, having taught it, and I don't know cancer biology as well as I know the others. So, sort of interesting. So if it's got embryology in it, that's fine. I can just go with that.
Yeah. Well, but you know, again, in kind of like the Levin work, CompuCell 3D would be good for cancer modeling too, in the sense of well, I'm not sure actually. So if it's a breakdown of multicellularity, well, again, I bet you he's got worked examples. I just haven't looked them up yet.
Yeah.
Oh, okay.
I mean,
So there's some Levin was doing cancer.
Oh, Michael Levin, yeah, has just been talking about how his work, you know, the planaria that he's working with, or that he has some of his examples are using, seem to be cancer resistant, or you know, are immortal and practically cancer resistant. Like they can you know, raise them in, you know, incredible toxicity, and you know, the animals seem fine. But in particular, his work seems to be focused on the role of electricity in multicellularity's collective intelligence, or, you know, collective in multicellularity socialism. Seems like communicates electrically, or, you know, what role it plays, especially in its spatial heterogeneity, differentiation. Yeah. Right. So, you know, like how you can play with you can play with some of the receptors and pumps to reconfigure an organism's spatial organization, right?
Well, it does have. I know epithelial cells run on calcium, just like muscles do, because it has a actomyosin ring.
Okay.
Right around
Yeah.
the top part of it, apical part of them.
Yeah.
Except in some exceptions, it's on the apical part of cells. It can apparently be somewhere else in some specialized cells, and it's an actomyosin ring.
Yeah.
It's non-muscle myosin, but it's still myosin. So you get cramps.
That's interesting. Yeah, I mean, so he doesn't just focus on those creatures, but he's even got like an active startup working on regenerating mouse limbs.
Oh, okay.
So
yeah, I need to have a look at that too. This is not for my 10,000-word essay. This will be for the confused professors, so they'll leave me alone later.
Well, I think it's definitely good for the the future applications kind of section. If you can relate your work to the things that Levin's doing, it's he's working on so many cool active research topics. This is the guy who put together with Josh Bongard the living robots, or is it Xenopus cells?
Yeah,
right?
the Xenobots.
Yeah, the Xenobots. Yeah. So again, just really, really interesting applications of really fundamental biology, right? And CompuCell 3D is great and seems to be a tool that lends itself to the kind of Levin-like simulations, but he's actually got some really cool software projects too, that really focus on the kind of cellular, non-neuronal cellular bioelectricity.
Okay. Does he get into mitochondria?
No, again, his work is very, I don't want to say abstract, because he'll do things that are so such incredible living demonstrations too, but the software is pretty abstract, or like Betsy, which is his electromagnetic simulator, 2D simulator, is still very, very removed from any kind of biology, but it's again, it's interesting demonstrations of I think he has it just because neuronal simulators aren't of interest to him.
No.
He wants, he's really trying to do something different.
I'm an electrical engineer by that's my foundation, so I'm more interested in the circuit and what's going on.
Right. And simulators like Neuron, which is an old school C++ package for doing reaction fusion simulations and allowing you to do kind of low level neuronal simulations. He knows that exists. So I think that's why he does things a little differently in his simulators. But his paper is worth looking at. He works in so many different areas. Is it Tufts? Yeah, Michael Levin.
He's at Tufts?
Yeah.
Oh, wow. Okay, I know somebody else at Tufts that I want to go there.
Me too.
And I met someone at Tufts. He has a nice program for doing simulations.
Oh yeah.
So we seem to be really into that. Okay, his name is Timothy, and I'm losing his last name here. Starts with an A. Anyway,
I just dropped a link for embryonic segmentation. I think with CompuCell 3D. You know, so like again, I mean, I was saying this Saturday. You know, the wealth of material from Glazer's group is just amazing. Yeah, he's got all his courses online, which I will say Josh Bongard does too. But yeah, Glazer's got all his courses online, all his student presentations, you know, the work they've done, as well as they do an annual workshop in summer school, you know, just to cover the software.
Okay, well, there's a program out there called Morpho.
Yep. Yep.
And that looked interesting.
If you've got a link, there's a lot of packages with Morpho as a name.
Okay. Yeah. All right.
If it's particular.
My only problem with it is I downloaded the whole thing, got everything downloaded except the last line of code, and it wouldn't download.
Mm-hmm. Well, I was saying to Lucas that one of the things I like to do, just to get my feet wet, so to speak, is package the software for Fedora, so that people don't have this problem. It's as easy as installing any other software. They don't have to build it. They just load the RPM.
For what? Fedora? Never heard of it.
It's like Debian for Ubuntu. It's like the
Linux.
Open, it's Linux, but it's the development version of Linux for Red Hat, or that's what the Fedora people would like to call it.
Okay.
And it was the 20th anniversary, so it's I'm feeling particularly old, and that's not when I started using Red Hat, but that's just when Fedora started.
Oh, I tried to put this on my lovely Windows machine, and the Ubuntu line, the last line for Morpho, it just won't take.
Okay. So send me a link. Where's this from?
Tufts.
Oh, it's from Tufts. Oh, wait. I think you did point me to this before. Wait. Yeah. Oh, right. No, it's coming back to me now.
Okay, well.
I just need my glasses. Yeah.
What's your email, and I'll send you what I know.
Oh, yeah, no. I'm on their GitHub now. Yeah. I've already got it saved. All right, small embeddable language for scientific.
Yeah, just seriously annoying. And I'm not very good with command line programming.
Right, and this is typical where it's like somebody's written their own tool, but that it's basically using BLAS, LAPACK, and sweet sparse in the back end.
Oh, okay. They're doing what?
Yeah, it's just yeah. Well, I think I had to look at this before, but yeah, it's all yeah. And this is the kind of stuff This is the kind of stuff that they were covering in this video that Glazer's group released, was playing with the code base and looking at the dependencies, what happens when you update the dependencies, what breaks, and how to merge that tree. It was very cool.
Okay, well, anyway, my computer doesn't seem to want the code, the Linux code. So
But this on and you're using Ubuntu?
yeah, it was Ubuntu.
Unjet.
Yeah, I don't know what that is.
Yeah. I'm familiar with a lot of obscure software.
Oh, yeah.
Scary software, yeah.
Well, that's great. Yeah. Thanks, Morgan, for the discussion about the CompuCell 3D.
Okay, I was just
Sorry,
wondering.
do you have a question?
Oh yeah, no, I was just wondering more about programming at Tufts.
Yes. I'm just looking at that now since my daughter seems to not want to go to school today.
Oh,
I'm not sure she has an actual excuse. I think it's just a preference.
okay, as long as it's not a reason.
Yeah.
Exactly.
As long as it's just a whim.
Right.
But yeah, and in terms of morpho, or
Yeah, sure. Morpho and other types of programs that deal with cells.
well, I mean, CompuCell 3D is the thing to look at. I mean, Morpheus is this other package that I was looking at.
Yeah.
And I'm pretty sure, it's from the High Performance Computing Group in Dresden, but I'm pretty sure it's because Dresden is the site for the German government's focused like splits, their Max Planck centers into specialties, and stem cell is Dresden's focus, or like one of the centers, Max Planck centers, is the stem cells is there. So it's doing. It's got a lot of worked examples. Yeah, CompuCell 3D and Morpheus.
CompuCell 3D is from Dresden?
No. That's from Indiana, and Morpheus is from Dresden.
Yeah. Morpheus.
Yeah, and
how about Morpho?
Yeah, Morpho I'm looking at now, and looks like like honestly, it's a lot like MATLAB in that it's kind of a language, a scripting language on top of BLAS and LAPACK.
On top of what?
Well, they're using numerical libraries that were developed by a lot of people a long time ago at national labs, mostly out of Tennessee, I believe. Oak Ridge. Yeah. Oak Ridge. Yeah. Okay, and it looks like they've got a make file.
You're looking at Oak Ridge?
No, that's just sorry, just giving you more detail than you need here.
Well, that's okay. You're looking at what?
Just going into the Morpho source directory. Morpho 5 is where they say to go, and then they've got a make file. Let's see if this runs. I don't know why it thinks it needs. Well, I just want to make. I don't want to install it.
Oh, well, that would be good too. I tried to install it.
Well, so I'd want to check this particular. I'd want to see. I like to see a file that before it requests, super user access.
Well, I was talking to Timothy Abernathy from Tufts, and I have his email. Like he was trying to help me with it, and like I said, it got down to the last line, and it just wouldn't take it. Like I'd type it in, and then it would erase itself.
erase itself. That's
Yeah,
pretty remarkable.
yeah, it has something to do with my computer.
Is that what he told you?
No, that's what I'm
Okay.
The computer is a little different.
It's not our code, it's your computer.
Yeah.
Yes.
Yeah,
That,
it could be both.
so many software engineers have said that.
Okay, all right. Well, it wasn't working. It erased itself.
That I can believe. So yeah, I need to see a little bit more about. I mean, it looks like you build two projects. One's called Morpho 5, which I assume is the language, and then you build Morpho View, which is obviously the viewer.
Yeah, it was Morpho View that I was having trouble with.
And they're both trying to put stuff into user local, which does take me back. That's quite old school these days. Like, yeah. This can easily be changed if a different location is preferred. So yeah, let me I gotta head out and get kids to school, but I'll have a look at this when I get back, and I'm sure we can get this built. But you say you're on Ubuntu?
Yeah.
Okay.
Okay.
But is that you're doing Ubuntu inside Windows?
Yes.
Gotcha. So your Windows subsystem for Linux.
Yes.
Gotcha. But the Ubuntu, like semi supported Ubuntu, yeah. Okay.
Whatever say, that's all I can do here.
Yeah, they apparently have an app specifically for that.
Okay, well, I did install it as per directions, and like I said, the last step on the view.
Maybe for the viewer.
Yeah, Morpho View. The last line wouldn't take. So how can I run the thing? This is like. I don't know.
Let me just.
The major problem with any programming that I do is installing the software in the first place. That is the main hang-up with it. I can usually figure out how to do it after that, but installing it, no.
Yeah. Well, this is and you're making a great case for why Linux packagers do the work they do. It's for exactly this problem.
Okay.
So yeah, I'm immediately getting a problem with the viewer. Oh yeah, GLFW. So yeah, I need to look at how old this project is, because I am running, at least I'm building this on Fedora. I've got an Ubuntu system that I use for just to keep things sane. But yeah, I'll have a look at this later and get back to you. Do I have your email, Susan? Or I can get it from Bradley.
I don't know.
Yeah. But happy to look at this. But curious of what you're hoping to get from it. Like, is it because they have like worked examples, or okay, all right.
Yeah, I want to put my design of epithelial tissue into it, and just and then run it from there.
But what do you get from that? I mean, in the sense of
I hopefully get the matrices that are behind running it, like some of the math, because yeah, I asked COMSOL, well, can I see some of the matrices that were produced? So they went,
Okay.
they laughed at me. All I got was a laugh.
Who laughed at you?
Anyway, I'm like trying to do a PhD here. I need the map.
And what tech support was laughing at?
Yeah, something like that.
Okay.
Yeah, it was during one of their presentations where you could ask any question.
Okay.
They just laughed.
Wow. That's coming from a place of privilege. If they're gonna laugh at you in front of
Okay.
publicly.
Usually when people laugh, I've asked an impossibly hard question. I've done that.
Well, in this case, it's just that they are a closed source. They're a proprietary company. Code base, and there are finite element solutions that are open source to give you that you can then get at any of the intermediary representations you want. But then they become much more, it becomes again like more an engineering project, potentially. This is yeah. Well, I'm curious what they're using here, but yeah, let me give it some attention when I get back.
Thank you very much. Thank you for.
I've got your email, and well, we've talked about Phoenix before. So this is FENICS. As that was there's a number of finite packages and communities where like
How do you spell Phoenix again?
Yeah, FENICS.
Oh,
So
okay.
yeah, and that's one that was what we used for our kind of finite elements electrical impedance tomography head modeling. But I had an engineer programmer working on it who was like a trained particle physicist.
Well, that helps.
Yeah. So and he was working with but he was getting a lot of advice and help from the community. And what's the other? I think it's GetDP. Is what. So like again, like I'm just drawing from examples in electromagnetic imaging, or electromagnetic head modeling. So there's a package called SimNibs, which is used for like TMS and TES modeling, and it's using GMesh and so that's GMSH and GetDP, which is another. I'm pretty sure GetDP's from a really good group in Liège. But these are used for modeling, electronics or electrical equipment. I think the GetDP developers won a Google Prize of like making their data center power systems like 10% more efficient or something like that. And, because like if you can make Google's power systems 10% more efficient, you save them like $20 billion or something.
Well, there's a lot to that.
Yeah. It's like
I know about building electronics and electricity left. That's the field I was working in, and as an electrical engineer, I know something about it.
I want to say I was saying all of that sight unseen, but is it? Yeah. It's definitely it's the GetDP guys that won the Google thing. But that's still very this is a super active project, right? it's a lot like the
How to stabilize your electrical system. For instance, when there's spikes coming into your system from your hydro, and Manitoba's hydro is spiky because it converts from DC to AC for the last step.
Yeah. So I've seen a lot of that kind of modeling and from the Julia folks actually. Modeling big electrical networks, power systems for states and countries and stuff. But anyway, I've got to take off. I will be in touch via email.
Thank you.
Yeah, no problem. And I dropped a link for Get DP.
Okay, yeah, I've got it. I captured it.
Yeah. They're great. All right.
Yeah.
Take care.
Thanks for
Thanks,
attending.
Bradley.
Bye.
All right. Talk to
Bye,
you later.
Susan.
Bye.
Bye.
All right, now I'd like to cover an additional paper, and this one is actually a theoretical paper on morphogenesis, but it ties into the epithelial paper we were talking about earlier in the meeting. This paper is called A Mechanism-Based Theory of Cellular and Tissue Plasticity. This is from a Chinese group in PNAS. And let's go through the significance section first. So the significance is that cells and tissues are known to undergo significant plastic deformations during many biological processes, and they list cell migration, tissue regeneration, organ development, and morphogenesis. So aside from morphogenesis, there are a lot of these plastic type deformations that occur. So these can occur in adulthood, or these can even occur in development, independent of morphogenesis. However, at this juncture, there is still a lack of comprehensive theory in the field. So they developed this mechanism-based theory that accounts for all key processes involved and achieves quantitative agreement with all existing experiments, which I don't think it's all existing experiments necessarily, but they kind of give they have to sell it somehow. Our theory not only provides a fundamental understanding of how plasticity is initiated in individual cells and then propagates within tissues, but it can also be used to guide the design of strategies for regulating biological processes such as tissue formation and embryogenesis. So this is, again, they're very well within what we've been talking about with respect to epithelial sheets and some of the sort of the movements and migration patterns in morphogenesis or in embryogenesis. So the abstract gives us a little bit more detail. So again, plastic deformations in cells and tissues, and that's how they're defining their area. So it's not just about morphogenesis or embryogenesis, it's about these plastic deformations. And so the fundamental question here is how plasticity is initiated in individual cells and then propagates within a tissue. So, the way they view it is that you have plasticity as a property of the individual cell, whereas maybe an internal property of the cell. That is, maybe it's a matter of gene expression or a matter of the cell's behavior within a sheet or within a collective of cells. And then those changes or plasticity propagates within a tissue. So it goes from a single cell to multiple cells. And so I don't know if that's necessarily a good way to view plasticity, but it is one way. So to state this another way, if you have a single cell, and let's take our epithelial sheet for a moment, we'll take a single epithelial cell here, and it has internal properties, which would be in, some sort of gene expression. So we might have a genetic regulatory network, or it could be the properties of the cell, the biophysics. The point is that they're talking about single cells. And maybe this is a case where you have a network of cells or, individual single cells, maybe in parallel. So you consider single cells, but in parallel. But cells are almost always embedded in some sort of larger system. So let's say that we have a sheet of epithelial cells. And each of these cells are operating independently. We'll just give them all sort of an internal model of some sort of plasticity. So, they might each have a switch where they go from one state to another state, where there might be a generalized plastic response that, is triggered in each cell independently. But as cells that are coordinated, say in a sheet, you have these coordinating factors. And they could be like signaling or some other, type of switch that would synchronize. So you have cell signaling and cell-cell communication, and those individual signals would become synchronized so that that plastic response is uniform across the sheet. And so that's maybe what they're arguing. That would be my sort of take on this argument, is that, plasticity may originate in individual cells, but it also needs to be synchronized in some way. And it's, it could propagate in the classic sort of agent-based sense or cellular automata sense, where a single cell has effects on its neighbors, and then those neighbors have effects on their neighbors, and so forth. But I think that you have to consider that sort of collective response. So this is, they say themselves, a mechanism-based theory, which is great. We need more mechanism-based theory, but we also need to link it with collective behavior type theories or emergent theories. I think mechanism-based and emergent-based theories are maybe, hybrid theories are probably the way to go here. So they talk about a mechanism-based theory of cellular and tissue plasticity that accounts for all key processes involved, including the activation and development of active contraction at different scales. So you have these different scales of contraction, active contraction, as well as the formation of endocytic vesicles on cell junctions. So, we talked about vesicles earlier in terms of being deformed due to different types of optogenetic stimulation. So certainly if you have cell-cell communication, the vesicles are going to play a role in that, and being able to sort of modify the conformational shape of those vesicles then plays a role in some of that extracellular communication or intracellular communication. So they talk about how their theory achieves quantitative agreement. That's important in theories, but of course it's not the only thing. Sometimes, we want theories that maybe don't agree with every piece of evidence because we want something that's overarching, and not every experiment's been done. So we want to have some open areas of inquiry. We want to be able to cast our net in a theory. We want to be able to cast our net very generally and explain not only the experiments that have been done, but those that are going to be done in the future, or even counterintuitive results that might change the face of what we know. So actually here they point out that, you can use optical or mechanical stimuli to sort of probe the cells, the myosin contraction and thermal fluctuation assisted formation and pinching of endocytic vesicles. So they focus on endocytic vesicles, could lead to permanent shortening of cell junctions, and that plastic constriction can stretch neighbor cells and trigger their active contraction through mechanochemical feedbacks, and eventually their plastic deformations as well. So let's map this out. I'm going to map it out, and then I'm going to show you the board. So what they say here is that I want to look at this, I want to underline this, draw this out, and then show you what we're talking about here. So they have some optical or mechanical stimulus, and this is of course a input to the cells, each cell. So every cell is going to have this input. And then we talk about myosin contraction. So there's a cell, you have vesicles on the sides. Those are being modified by this stimulation. You have a cytoskeleton, which is being modified by the stimulation. You have a cell modification, and so you have these, and so this is a permanent shortening of cell junctions. So these cell-cell junctions that happen between cells, you have a shortening, a modification of the cell junctions, and then plastic constriction can stretch neighbor cells and trigger their active contraction. So you have opposing forces between cells, and then you have another cell. I'm going to draw 2 cells here, and just kind of in a sheet of cells. Through mechanochemical feedbacks, so these modifications are feeding back on each other. Eventually, they're plastic deformation as well. So this is my drawing here. It's not very good, but you can see that you have the stimulus coming in. You have these cells, a neighbor cell, which is kind of half shown, and the main cell, which is down at the bottom. You have all these sources of modification. You have the sheet of cells on the left with feedback, and those are the mechanochemical feedbacks. So this process on this cell down here is happening in every cell simultaneously, or maybe not simultaneously. That's, maybe a thing that you think about in a theory where, you would make the assumption, say, that everything is happening simultaneously, but of course, it may not be happening simultaneously. It may be happening in a way that's asynchronous, and so you have to consider that as well. But you have these general mechanochemical feedbacks, which are, of course, it seems like magic, right? I mean, you say something is a magical feedback, and, it's obviously now a collective behavior. But these feedbacks, work at different scales and in different ways. So you might, for example, stimulate the cell. You might change the shape of the vesicle. It might affect gene expression or cell-cell communication in some way. You might modify the cytoskeleton in the actin filaments in the cytoskeleton, and it modifies the shape of the cell, which can modify the cell, the gap junctions, and other types of interfaces with other cells. And these can happen differentially across the sheet of cells, so that when you have a sheet of cells, and we'll go to, I'm going to draw a sheet of cells here. These are these epithelial cells, that they have these like properties along an axis that result in maybe some sort of critical event that happens. So for example, if you have enough forces going in one direction, you might end up being able to slip a cell out. The cell might start to come out in this direction, and these other cells might end up coming together. There are other types of collective behaviors that might happen. For example, you might have, deformations where the sheet splits in two, or other types of things that might happen. So there are all sorts of things we can use sort of a mechanistic theory to explain. Now, whether the mechanistic theory is the best way to do that is, of course, up to the reader to decide. So this is kind of what they're saying. Their theory predicts that endocytic vesicles with a size of around 1 to 2 microns will most likely be formed, and a higher irreversible shortening of cell junctions could be achieved if a long stimulation is split into multiple short ones. So they're really getting very mechanistic, I guess you could say, and they're kind of predicting these very small components and how they contribute to larger cells, and then ultimately the behavior of cell collectives. And so all in quantitative agreement with experiments. So they come up with this theory based on experiments, but also based on sort of, what they think is the optimal size for this. Our analysis also shows the constriction of cells and tissue can undergo elastic unratcheted to plastic ratcheted transition as the magnitude and duration of active contraction increases. So as there's contraction in the cell, you have these what they call ratcheted transitions. And ratcheted, at least as far as I understand it, and there are different types of ratchet mechanisms that theorists have used. There's, of course, the Mueller's ratchet, and there are other types of adaptive ratchets where basically change happens in one direction. So a ratchet allows you to sort of move things in one direction and then locks out movements in the other direction. So you move a ratchet, you kind of move things forward and then it locks, and then you put it back on and you move it forward and it locks. So it's this one-way process. And so elastic, in their estimation, is unratcheted, which means that elastic changes can be, they don't have any directionality. So things can stretch or things can expand, things can contract, and there's no long-term memory for that. There may be some hysteresis in the system where things get sort of, things can return to their, can't return to their original state. But this is not directional. It's not meant to be directional, as opposed to plasticity, which is ratcheted, which means that something goes in one direction. So plastic responses often are adaptive in a way that improves, say, like the dynamic range of the cell, or it improves the sort of increases in size or decreases in size. There's a directionality to the change. And it's not just simply hysteresis, where you have this, it can't return to its original state. It's very much intentional to provide an advantage to the cell and maybe even to the organism. So they say that there's this sort of transition from elastic unratcheted to plastic ratcheted transitions as the magnitude and duration of active cell contraction increases. So as active contraction increases, this magnitude and duration, then there's this transition, ultimately resulting in the propagation of plastic deformation waves within the monolayer with a constant speed, which again is consistent with experimental observations. So there are these plastic deformation waves. So plasticity is propagated throughout the monolayer, and you get these changes. So they point out that tissues, organs, and organisms assume complex three-dimensional geometries throughout morphogenesis, that large numbers of cells deform and move in a highly coordinated and programmed manner. Mounting evidence has demonstrated the capability of cells to undergo irreversible deformations, individually or collectively referred to as cellular tissue plasticity. So they define this sort of ratchet, this plasticity ratchet. Mechanistically, plastic deformation of cells involve various events across different length scales. So at the subcellular level, you have lipid vesicles that form and then pinch away from the cell membrane. So they kind of pull away, and this is what they call endocytosis, effectively resulting in a plastic strain in cell surface. So as these vesicles form, they kind of pull on the surface of the cell, and they do different types of deformation. And then you have things going on at the tissue level. The plastic contraction of individual cells can activate collective contraction of their neighbors, cooperative constriction, something that cannot be achieved by elastic cellular deformation or competitive constriction. So this is something that where you have this collective contraction versus just simple cellular deformation, things happening in different cells competitively in the sheet versus things that where the cells are cooperating to have this sort of collective movement. This eventually initiates molecular and deformation waves within the tissue, triggering its morphogenesis, leading to the formation of spatial patterns. So they talk about 32 and 33, and I'm interested to know what these molecular and deformation waves are that they're referring to. So citation 32 and citation 33. This one here, 32, is genetic induction and mechanochemical propagation, a morphogenetic wave. And we've talked about morphogenetic waves as something that maybe needs to be viewed critically. Then 33 is control of osteoblast regeneration by a train of ERK activity waves. And these are electrochemical waves that occur in, so this is an expression of ERK, and this is an activity wave, trains of activity waves that are associated with that. Then, there's some other nice citations in here. We should go through these citations at some point. This would be very useful. Let's go to the figures. So figure 1 here shows kind of what they're talking about here. Here are your vesicles. And I drew them on the outside of the cell, but they can happen, within the cytoplasm. They come up to the surface and they sort of deform the cell in different ways. And so this is where the vesicle itself is regulated in terms of its size and how it's pinched off by these dynamin molecules. And here are the actin filaments in the cytoskeleton, which can also push on this vesicle. Then you have one plastic cell in a sheet of cells. So this plastic cell can do its own thing, which is competitive, or it can act in concert with other cells, which is cooperative. And so this is an example here of things happening at the tissue level, the cellular level, and the molecular level. Again, their focus is on mechanism, but they're kind of generalizing to collective behaviors, which may or may not be particularly useful if you're trying to understand things at multiple scales. This shows where you have mechanical stimulation by strain or optical stimulation by light, and these activate signaling proteins inside the cell. So this is at the nanometer scale. At the micron scale, you have these different forces acting on the cell. And then at the millimeter scale, you have these sheets of tissues where you have multiple cells interacting at their interface. So this is the tissue level. You have these aspects of cellular mechanics. You have at the cellular level, you have plasticity mechanisms, membrane tension, and vesicle formation. Then at the molecular level, you have signaling molecules, molecular recruitment, so recruiting other molecules into macromolecular complexes, and then contraction. And so this all contributes to cell strain. So this just shows how cellular plasticity acts across scales. And again, you need some sort of multi-level model or some sort of hybrid model to treat this in terms of modeling and in terms of even understanding the problem fully. Figure 2 shows modeling contraction and thermal fluctuation assisted formation of endocytic vesicles. So you see the vesicles here where you have this, the length of this surface element here in B at the top. Then the next image shows bending. So there's this bending at an angle, it subtends at a certain angle, and it pulls up the sort of the edge of the vesicle. And then the vesicle forms as this semi-independent thing where it's tied together by the dynin at the base. And then eventually this vesicle gets pinched off. So the dynin pinches off the neck of the vesicle and it floats off as something carrying molecules or something else and moving away from the cell membrane. In vesicle breaking, that's the dynin that breaks apart and the vesicle sort of gets unbound and then shrinks back into the membrane. So these are dynamin, serves to sort of regulate this process. You have all these potential vesicles on the edge of the cell. They break off and move inward, and they're involved in signaling, but they can affect the shape of the membrane. So you can see this where you have this active process of dynamin, the actin filaments, and the vesicles all acting together. So you have this process of formation, you have this process of stimulation. Okay, so a lot of this paper is focusing on the mathematics of sort of cellular physics, and it's talking about the formation of vesicles. It's also showing some of these stimulation experiments where you stimulate some cells in the center and they affect cells on the periphery. So you have these illuminated cells, unilluminated cells, and then this boundary stress of the community of cells. So a lot what we saw in the paper earlier in the meeting where we have this focus, we have light stimulation where we focus a light in a certain place and it has an effect. In that case, they were using optogenetics. In this case, I think they may also be using optogenetics. but you can actually have different types of light stimulation independent of optogenetics. The control isn't necessarily there, but it may have an effect on the cells with respect to the other cells. And so they show some graphs here. So they trigger something by light at the 0 minute mark, and then they show the dynamics of these different things. So our graphs are, we have simulated length evolution of junctions under a single 5 minute, 10 minute, 20 minute, and 40 minute length. So this is where we have 10 minute, 20 minute, 40 minute, and 60 minute intervals. And so you can see that there's a little bit different time course where if you increase this window of exposure, so from this in B it's 5, so you can see the shaded area of 5 minutes where there's this junction length that shortens and then rebounds. And of course, as it rebounds, it does not exhibit hysteresis as it retains its length of 1. And then in C, you have this stimulation of 10 minutes in the shaded area. So you have this stimulation process, the junction length goes down. But then there's significant hysteresis after the light stimulus is removed. With 20 minutes, you see a similar result. And then with 40 minutes, you see a similar result where there's this significant hysteresis and non-recovery. And it's, actually the junction length shortens significantly in this case. And then in F, you have this pulse of 20 minutes and 20 minutes where you see this, where if you pulse it over time, you pulse it, you have a light pulse, you remove the light pulse, you add the light pulse again, then you remove it, you get this serial hysteresis where you have a junction length of 1, it decreases to about 0.65 or 0.7, it rebounds to about 0.8, so there's some rebound, but there's still hysteresis, and then you stimulate it again, and there's hysteresis on top of that. So you have 2 hysteretic events that occur in a staggered fashion, and it really can reduce the junction length. So light stimulation can affect these factors. And so then in G at H, you have normalized myosin activity over that same time period. So myosin ramps up as the junction length decreases, and then myosin length actually exhibits no hysteresis because it rebounds over the 20 minutes back to its original activity of 0. And so, it's not stimulating any background myosin activity. Then the junction tension, which is in nanonewtons, you can see that there's this drop with the treatment, there's this kind of noise, and then it drops off, and then it rebounds to its original. Actually, the junction tension increases a little bit as the treatment is removed. And this is in the pulsed regime. So you have these pulses that decrease the junction tension, but the junction tension rebounds. So this is just some example of how they can break down a single process and get measurements of it and say things about it. So this is showing a phase transition during the initiation and propagation of cell plasticity in Figure 4. And these graphs are kind of showing the phase. They're phase portraits of these different variables. So contraction amplitude versus normalized time. You see that there's this plasticity that increases as contraction amplitude increases, but also over time. And then the contraction amplitude with time, there's this adjacent cell contraction. So you have a central cell, and this figure D, subfigure D shows this. So this is the central cell in this purple, and this the adjacent cells are in sort of this lighter color. And so what you have basically is the central cell has this plasticity, and the adjacent cells are doing things in response to that. So in the competitive mode, you have single cells that are interacting with its neighbor cells, and they're influencing their neighbor cells their neighbor cells may be responding to that, whatever the central cell is doing, or it might be reacting to it in another way. In the competitive mode, it's basically the central cell. That's the main driver of everything. In the cooperative mode, you have adjacent cells, which can be the drivers as well. And so they're cooperating versus competing. And so you can imagine that all these cells can be central cells at the same time. They're just in a different mode of interaction. They may be competing with one another as opposed to cooperating. And so you can see that in this case, you see that over time, the contraction amplitude increases in the central cell, the plasticity increases, and this has an effect on the adjacent cells. So the adjacent cells will contract, the contraction amplitude will increase over time, and the adjacent cell plasticity ultimately then will increase over time because it's being forced by the central cell. In E, you see that there is these ratcheted and unratcheted regimes. So with contraction amplitude, over time, initially it doesn't have any effect. It's uncontracted, so it's just kind of neutral. And then over time, you start to get these amplitudes where you have this unratcheted regime. So it's kind of perturbed, and it's responding to that perturbation. Ultimately, though, this is a ratcheted form of change. So you get this directional change, and it's really with high amplitudes. Over time, that amplitude can be lower. It can be almost a little bit above 0.5, but you can see that there's this process of it being unaffected versus having this almost like Brownian noise going on to some sort of directional ratcheting of plasticity. And so that's what they're doing here. They're talking about this phase transition where you go from like a state of quiescence to a state of sort of response or reaction to this then state of plasticity. And so this figure talks about propagation in a tissue. So all these hexagons are individual cells. And so you can see that they've simulated this where over 25 minutes, where you have a parameter cell tension in nanonewtons, and it's basically propagating out from a central cell or a central set of cells. So you can see at 0 minutes, we have nothing going on. At 2 minutes, the central cell tension in the central cells is lower than the surrounding cells. And then as we move through time, that area spreads, so the cell tension drops. There's cooperative interactions going on. And over 10 minutes, 18 minutes, and 25 minutes, at 25 minutes, almost the entire sheet has lower cell tension. And the center actually has a little bit higher cell tension than the edge, the leading edge here. So there's a propagation along the front that's concentric and spreading outward. And so that's what they're showing here. So they conclude by talking about how this model can be applied. So although by revealing the mechanisms by which mechanical plasticity is initiated and propagated in cells, so now they're very specific in talking about mechanical plasticity. So it's not just about generalized plasticity, it's about this mechanical plasticity. This work enables a major advance in our understanding of important biological processes, such as tissue formation, wound healing, collective cell migration, and embryogenesis. So it's not just developmental, it's any kind of plasticity, but it's mechanical plasticity that they focused on here. In addition, the model developed here may also provide critical insights for applications like the formation of synthetic extracellular vesicles and organoids. So they throw organoids in there as a way to sort of generalize this theory. We talked about organoids earlier in the meeting and in other meetings where there are these challenges to modeling organoids. The question is, are organoids like a developmental system, or they maybe have their own properties that aren't necessarily like a developmental system? In organoids, you're often dealing with cells that transition from some sort of stem cell-like state, some pluripotent state, to a certain cell type. But those cell types aren't necessarily the same cell types that you might find in biology. So it's an important point to think about. Finally, by incorporating more features such as the plasticity-induced curvature of cell layers, so they didn't include curvature in these models, it's just like a flat sheet of cells, as we saw with the epithelial cells, anisotropic boundary conditions of tissue, so they're different conditions at different portions of the sheet. So you might be like on one side of the sheet versus the other. There are different boundary conditions. And reversible pinching and breaking of vesicles, which, you can have this pinching off of vesicles and they might float away, but they might also come back down, redock, and kind of be resorbed into the membrane. These sorts of things might happen as well. It is conceivable that the present theory can be extended to studying interesting phenomena like three-dimensional structure in tissue, pattern formation of cells, and the hyperelastic response of active materials. So this is all kind of how they're generalizing their theory. So thank you for paying attention. I hope you learned something.